Hello, my name is Jalloul and I'm currently a postdoctoral fellow at Germline lab at Rutgers Cancer Institute. The poster I'm presenting is entitled Integrated Inference of Specimen Purity and Mutational Status in Clinical Tumor Sequencing to Inform Treatment Strategy. To start off, I'll explain some of the objectives driving this work. Recent advances in clinical sequencing technologies have resulted in unprecedented access to the genomes of individual tumors. However, most clinical-grade implementations of sequencing lack patient-matched germline DNA, and germline testing becoming a standard of practice in oncology might still be a long way away. Because of that, most cases require further analysis to infer specimen tumor content and to infer the mutational status of variants characterizing the tumor. Given, for example, a potentially pathogenic mutation detected in a tumor suppressor gene, it is imperative to determine whether it is germline or somatic and resolve any evidence for the loss of wild-type copy, which may be pertinent for treatment efficacy and determining the effect of the mutation, for example, if it results in a complete loss of function. Genomic heterogeneity in both tumor and non-tumor cell population also confounds distinguishing subclonal tumor alterations from those possibly originating from non-tumor components in the tumor microenvironment. Therefore, as the use of tumor-only sequencing assays increases, systemic interpretation of clinical-grade data is necessary to accurately describe the genomics of a single tumor. Here we present a set of integrated bioinformatics tools to interpret mutations in an individual tumor sample. The information theoretic algorithms are ALLFIT, or allele frequency-based imputation of tumor purity, which is developed to estimate tumor specimen purity and its associated confidence intervals based on allele frequencies, or VAFs, and focal copy numbers of detected variants, which may be heterozygous, under LOH, or amplified at their loci. Logic, or loss of heterozygosity inference calculator, is developed based on this model selection scheme to find the most consistent model for mutations, germline versus somatic status, and infer LOH and mutated alleles copy number, incorporating VAF and purity confidence interval. Figure 1 shows a schematic of the processing methods as well as the detailed explanation of both algorithms, which are mostly based on modeling the likelihood in a binomial distribution, calculating a KK information criterion weights, which can be used to compare the likelihoods, and can be used by logic as likelihoods for mutational status. We evaluated ALLFIT using a comprehensive set of simulated datasets that included somatic and germline variants under different mutational models while assessing the impact of subclonal and highly amplified. A description of these cohorts is listed in Table 1, seen here. In general simulations, ALLFIT performed with 92% accuracy, outperforming other commonly used algorithms such as absolute. For simulated data comprised of both clonal and subclonal mutations with varying ploidy, ALLFIT's purity estimates corroborated with simulated values in 86% of cases, with Pearson's correlation coefficient at 0.99 compared to absolute's agreement in only 35% of cases with R equals 0. We also applied ALLFIT to clinical sequencing data of 1,861 solid tumors collected at Rutgers Cancer Institute, including 199 specimens harboring one hotspot mutation in P53 and 73 cancer specimens with 1 to 2 pathogenic APC mutations. For the hotspot P53 mutations, which are expected to be under LOH, copy-neutral LOH, and indicative of tumor content, we observed corroboration between the estimated and accurate purity values with a correlation coefficient of 0.51, while for pathogenic APC mutations, which could either undergo somatic LOH or somatic copy-neutral LOH if there is one APC mutation, or they could be individually somatic heterozygous if there are 2 APC mutations, we observed a correlation coefficient of 0.6 with purity estimates. Next, we evaluated our integrative pipeline for identifying germline variants detected in tumor-only sequencing using the data from 1,631 adult cancer patients consented at Harvard Cancer Institute. In this cohort, 427 patients had 676 variants detected in high penetrance cancer predisposition genes: BRCA1, BRCA2, MLH1, MSH2, MSH6, and PMS2. Comparison of inference results with germline testing data showed an overall accuracy of 89% and 88% using histological purity and all-fit purity estimates respectively, excluding the ambiguous patients. Clustering of predicted germline and somatic variants is consistent with expectations based on VAF and purity estimates. However, certain caveats remain in properly identifying the mutational status, which is reflected in the ambiguous calls around 50% VAF and purity. When interrogating the false predictions for all genes, it is apparent that most falsely identified germline variants have VAFs higher than 50%, and falsely inferred somatic variants have VAFs below 50%. This suggests computationally accurate predictions, which calls for further investigation of the reported mutational status. In conclusion, note that despite the limited breadth of clinical assays that often interrogate only a few hundred genes, simulations show that their sequencing depth of 300x provides sufficient power for all-fit and logic to distinguish mutational models and to infer LOH. Our analysis may be confounded by the absence of variants from different mutational models, presence of reversion mutations, poor specimen quality, or low sequencing depth. Overall, this work demonstrates the need for a systemic effort to interpret clinical-grade sequencing results and shows that utilizing an interactive presentation of molecular data for clinicians for accurate inference of mutational signatures and dissection of heterogeneity can generate diagnostic hypotheses that, when clinically tested, lead to improved prognostication, ensuring that treatment strategies are correctly focused on tumor alterations. Thank you for listening.